So, thank you. So, can I have the light be down? Thanks. So, if you coming down on this symposium on microbial complexity, and so if you look in the environment, I think it's quite easy to recognize that, when you look at bacterial population in the environment, so most of the complexity comes from the fact that very often, not always, but extremely often, you actually find these populations are a complex population, usually attached to each other or to surfaces that are called biofilms. And while this community have attracted a lot of attention for different reasons, because they form in all environment and they play important ecological roles, but also because they impact entropic systems, including, they are cause of infections when they are caused by pathogenic bacteria, or they also are involved in biofouling. A lot of people have been trying to identify what makes the biofilm so special, or what are the activity of these communities, and how this activity can actually be different from those of the free-floating bacteria called planktonic bacteria. And so one of the specific functions that can develop within biofilm are essentially, very often due to a very peculiar environment that is created by the formation of this community itself. So very rapidly, if environment allows it, you have formation of a thick community in which gradients can be formed. So diffusion gradients, and this has a lot of impact on, for instance, what could happen between bacteria in this community, as beautifully exemplified this morning by Kevin, but also gradients of oxygen, of nutrient, of waste. And this creates a physical-chemical heterogeneity that is thought in the field to be at the origin of the creation of gradients. And gradients that are very complex within this community, so you can create micro-niches, and which allows for the possibility of occurrence of local differentiation that would actually potentially lead to community-associated properties. And a lot of people have been trying to answer this question: what do bacteria do within these communities? And so the take we have been trying to use to go after this question is to try to identify metabolite and secondary metabolite that would have been mostly or more particularly produced within these environments. The problem with that is that if you try to actually go after the diversity of the metabolite that you can find in this environment, you realize rapidly that this is extremely complex. A zoo of small molecules that are produced at different rates by different type of population in different environments, and this is very difficult. So what we've been trying to do is to try to reduce this complexity. And to do that, we focused our attention on a specific class of secondary metabolite that have properties to be volatile and to be released in the bacterial headspace. And assuming that by doing that and only concentrating on these volatile compounds, you would actually have a way to, while not looking at the whole diversity, look at some and potentially identify some of these molecules that could be produced in these communities. So these volatile compounds have some role in communication and airborne interaction with the bacteria, but what I would like to focus on today is, and also to see how looking at this volatile could also give us insights on what is going on in terms of metabolism in this community and how widespread it could be. So the way we perform our experiment in the lab is we produce biofilm in a sort of continuous flow culture where you have surfaces on which biofilm can form. And in this condition, bacteria are removed by the flux, so you mostly have biofilm formed and very few planktonic bacteria. And you can put this biofilm biomass in small vials that have been designed to actually study volatile compounds. They are specifically adapted for solid phase microextraction, so meaning that you can actually have the biomass here, and then if there is any volatile produced by the biofilm, they can be released in the vials, and then these vials can actually be subjected to analysis with small cartridge, SPMA cartridge, and in which you can actually trap the volatile compound and analyze it. And if you do the same experiment with the same bacteria, but grown not as biofilm, but in planktonic culture, where bacteria are planktonic, isolated, and where the medium is supposedly homogeneous, you can do the same analysis and do a compare and gas chromatography and mass spectrometry analysis to actually identify what could be the difference between these different environments. So, and sure enough, even if when you do this sort of analysis and you compare the nature of the volatile compound that you can detect in these conditions, you find also a zoo of peaks that are or not there between biofilm and planktonic bacteria. But what I would like to discuss today is the fact that you can identify some peaks that are extremely similarly specific to the biofilm environment. And for instance, we identified one peak here produced by biofilm from the E. coli K12 strain, and I will talk about other bacteria a little bit later. And this peak is actually produced in mature biofilm at 24 hours. But interesting enough, and this is exactly what we wanted to actually identify, it is not produced in planktonic bacteria, whether it's 24 hours, but also almost cooked 48 or 72 hours long planktonic culture where bacteria are very, very far into late stationary phase. And suggesting that this molecule is produced in biofilm condition and not in the planktonic condition we have been using. So we use rich medium, and I'll come back to that. And so in this condition, you can also follow that this peak here could actually increase over time if you actually stop the experiment later in the biofilm development process. So at 16 hours, 20 hours, and then you have a very large increase of the molecule if you wait for the biofilm to grow older. So suggesting that this is proportionate to the extent of biofilm development. So mass chromatography told us that this molecule is 1-propanol. So it was a surprise, because if you look into all metabolic analyses that have been made so far, and for like more than 75 years on E. coli metabolism, no propanol, so 1-propanol equal propanol, no propanol can be found in E. coli. And to such an extent that people have been trying to introduce propanol capacity into E. coli, using it as a workhouse. And the reason for that is because propanol is a widely used industrial solvent, and it's also a very good biofuel compared to ethanol, if only we could make it in large quantity. And so people try to take some genes coming from bacteria that were producing a little bit of propanol, such as Clostridia or methanococci, and introducing them in E. coli, and shutting up some metabolic flux in order to introduce in E. coli the capacity to make this molecule. So this suggested that maybe what we actually stumbled into during this compare analysis was a metabolic pathway that could actually be specifically activated in biofilm conditions. So the conditions leading to propanol are readily analyzable if you actually study how much propanol you can make in fermenter conditions. So this is, for example, the black peak here. But you can actually, if you do this biofilm in this fermenter in totally anaerobic condition, you have the exact same amount of propanol which is produced. And, but if you, instead of making biofilm in this condition where very rapidly, it's not fully anaerobic if you do that normally, but because of the depth of the biofilm, you rapidly have accumulation of gradients of oxygens. But if you do that in a very different way, and you have a small movie here where you do biofilm in hyperaerobic conditions, so in this case, you bubble into the fermenter air, and so the biofilm is formed the same way, but in this condition, you have almost no propanol made, suggesting that the aerobiosis inhibits propanol production. So you can, if you force planktonic culture into anaerobic condition, you can make a little bit of propanol, but you never have the high biomass which is actually produced in biofilm condition. In this condition, you always have a huge number of bacteria that is exactly at the right concentration, anaerobia or very, very low microaerobia, which leads to the induction of a metabolic pathway that would lead to propanol production. And these conditions are extremely frequently found in all sorts of multi-species environment and biofilm that you can find either in soil, potentially in gut, but also in all type of biofilm that you can analyze. So what is the pathway? So we assume that in order to start somewhere, we assume that in order to make this alcohol, you have to have somehow an alcohol dehydrogenase involved in the process. And actually, if you look into how several groups try to introduce propanol capacity into E. coli to make a little bit of propanol, they always start by adding an alcohol dehydrogenase coming from Clostridia or from methanococci for that matter. And so we looked into E. coli, into known and potential alcohol dehydrogenase, and we systematically inactivated the corresponding genes. And what we found, looking at doing a GCMS analysis here, is that one of these alkyl dehydrogenase, ADHE, is actually, when inactivated, render the bacteria unable to form biofilm in any known conditions, so, and to have no propanol made in biofilm condition, suggesting that this pathway involves somehow ADHE. So ADHE is a very, very interesting enzyme. It's a multifunctional enzyme. There is at least 3 functions, so I will only describe 2. And so it's a bifunctional enzyme because it takes an acetyl-CoA, and through acetyl-CoA acetaldehyde dehydrogenase activity, transforming it into an aldehyde, acetaldehyde, and then it also has an alkyl dehydrogenase activity, so transforming this aldehyde, acetaldehyde, into ethanol. And I will come back to this 2-step activity in a few slides. So we had a pathway in which this ADHE enzyme was involved and could contribute, and we tried to analyze the pathway by looking at what could be the more upstream precursor of the pathway that would lead to propanol production. So looking at all the possible pathways that we could think of or gather from all organisms, we tried to look for precursor, and so what we found is that among all the carbon sources and different precursors that we could add, only adding the amino acid threonine would actually lead to an increase. And here I changed the scale of the peak I've been showing to you so far, because if you add threonine to a 24 hours biofilm, or you form biofilm with threonine and wait for 24 hours, you have a very, very sharp increase in the quantity of propanol which is made. And this increase is ADHE dependent, because if you actually do that in an ADHE mutant, which show no defect in biofilm formation, you have no propanol made, suggesting that the precursor of this pathway is threonine. And this has been confirmed using labeled threonine, for instance, and looking at how the carbon is actually transferred by NMR, and so all the labeled threonine goes into propanol. So we have a pathway where threonine is the precursor, and what are the other components of the pathway. So we knew that somehow propanol to be made comes from threonine degradation. And if you look into, again, the metabolism, there are not so many ways threonine can be degraded. And in prokaryote, these are the 4 ways. So we knocked out the genes going into the propanol pathway. And while the different pathway leading to acetyl-CoA, which could have been a route, or methyl glyoxyl or the branch chain amino acid would actually had no impact on propanol production, if you actually inactivate a gene called TDCB, which codes for an enzyme called threonine dehydratase, had been thoroughly studied during early work on allostery, but also pathway leading to amino acids such as valine, leucine, and isoleucine. If you inactivate TDCB, then you actually knock down the capacity to make propanol in biofilm without touching the propanol or the biofilm capacity. And so suggesting that somehow the pathway leading to propanol was in the vicinity, let's say the metabolic vicinity of the pathway that would involve TDC and maybe the pathway leading to propionate. So what is TDCB? So TDC stands for threonine degradation. So this is a pathway that involves an operand which is described here, and this TDC operand is first induced by threonine. Actually, in the operand, you have a gene coding for transporter of threonine that imports threonine into the cells. Then the different genes of the operand codes for enzyme that are located on the pathway that goes from threonine to propionate. And so the TDC operand then is induced by anaerobic conditions. So they have 2 inducers, threonine, availability of threonine, and anaerobic condition. And if you actually compare how these genes are induced, if you compare gene expression between planktonic condition or biofilm condition, you also see that you have a very sharp induction of TDCB and in some extent of the other genes, but not some of the other pathways here that I will describe in a second. So the TDC operand is then also induced in biofilm condition. So, however, this pathway, that you have here, leads for threonine to propionate. And how is it that this pathway somehow could actually lead to propanol production? So, in order to clarify that, we systematically inactivated all the genes of the pathway. So, we already know that if you knock down TDCB, you have no impact on growth, but you have no propanol. And the other genes, like for instance this one, ELVA and PTA, and so PTA is here and other genes there, if inactivated, they leads to no impact on propanol production, and they are mostly involved in the aerobic synthesis of propionate. So you have a pathway which, if you only take into account the gene that affects propanol production, include TDCB and TDCE, but no other genes, suggesting that somehow the pathway we're after would actually branch out from the propionate pathway, maybe at the level of propionyl-CoA. And so this is how, knowing that the ADHE is a promiscuous enzyme that has multiple functions, could come into play. Because as ADHE is able to actually transform and convert acetyl-CoA into acetaldehyde, we showed that propionyl-CoA ADHE could transform propionyl-CoA through coenzyme A-dependent acetaldehyde dehydrogenase activity, it's a mouthful, into propanol. And then propanol, like acetaldehyde, so propanaldehyde, could actually be converted into propanol by the alcohol dehydrogenase activity. And so we have many evidence, but for instance, if you actually add to a wild-type propanol-producing biofilm, you had propanaldehyde, then you have a sharp increase in the amount of propanol that can be made, indicating of the conversion of propanol into propanol by ADHE. And this increase is ADHE dependent. So what we've been showing, what I've showed to you so far has been done in E. coli K12, a biofilm-forming strain of E. coli K12. So is this ability to divert the pathway to propionate into propanol a widespread capacity? And so first, we actually tested a lot of different E. coli, and as you can see here in the sub-panel, so each time you actually, all tested E. coli are able to form by propanol in biofilm condition, while if you actually test that in planktonic condition, they do not, so, and indicating that the production is detected in all tested E. coli. But if you actually look into the diversity of the genomes, and you actually, instead of testing them systematically, you actually look for the co-occurrence or good homologs of the genes that cause for the enzymes involved in the pathway, so namely ADHE, TDCB, and TDCE. You can see that, the darker it is, the stronger is the homology, that most of the strong homologs are actually found in gamma proteobacteria, and especially in Enterobacteriaceae. And consistently, many Bacteriaceae also have this capacity to form propanol. Here it's Salmonella, Shigella, Citrobacter, but not all. So you find it in a vast array of bacteria that can, Enterobacteriaceae, that can produce propanol, but not all. So what could be actually the function of being able to produce propanol? So at first, propanol production is not, propanol itself is not used as the carbon source. If you actually use culture or biofilm made in a minimal medium and you actually add threonine, you have a huge increase in propanol. But then if you add propanol as a carbon source to another bacteria, it won't actually help it to grow. Moreover, we very thoroughly try to see and detect any function associated with the production of propanol on other bacteria, and we could not detect any toxicity. Obviously, propanol could be toxic at very high concentration, but at physiological concentration, whatever quantity of propanol is produced in the biofilm or in the culture, in anaerobic culture, you have no toxicity. So what could be the function? So when you look at this new pathway that branched from propionyl-CoA to propanol, you also see that this conversion is actually require another conversion, which is a very important conversion. It's a conversion, a reoxidation of a redox cofactor, nicotinamide adenine dinucleotide, NAD, which is extremely, which is under two forms, the redox cofactor, the oxidized form of this redox cofactor, NAD, and the reduced form of this cofactor, NADH. And all metabolic reactions almost need NAD to actually process into the further reaction. And what happens in metabolism is that this NAD is reduced in NADH, and in order for the reaction to continue, this NADH needs to be restored and recycled and reoxidized into NADH. And if you actually look into this reaction, you have two molecules which is reduced per molecule of propanol, suggesting that this pathway is likely involved in reoxidation of NADH into NAD. And so if you actually compare the ratio of NADH onto NAD in the wild type and on mutants that are affected for propanol production, TDCB or ADHE, And then you can see that you have increase in this ratio, indicating that you have more NAD plus made in the wild type than this mutant. And you have about a 6 to 10-fold ratio of NAD produced in the wild type compared to these mutants. But if you inactivate the pathway into at a place where it doesn't affect propanol production, for instance, if you inactivate TDCD, it has no impact on propanol production, then you have no impact on this ratio of NAD and NADH. So this pathway has all the making of what is classically described as a fermentation pathway. And so what we propose is that this pathway could actually contribute to the general redox balance of the bacteria that develops it. So, as I say, NAD is transformed and reduced in NADH by general metabolism, and in presence of oxygen, NADH is reoxidized in NAD without any problem, or in presence of another electron acceptor by anaerobic respiration, you have recycling of this NADH into NAD. But in absence of oxygen, or in absence of alternative electron acceptor, so what comes into play is a process called fermentation. So if you have fermentable carbohydrate, this somehow at some point leads up to pyruvate, which can be reduced and into different products, which along the way allows the reoxidation of NAD. And so this allows the metabolism to continue. But what happens if you are in an environment where you have no sugar? What happened to life when there is no glucose around, you know? And so this is a situation that you can find very easily in all kinds of environments where biofilm are present or in the gut. And we propose that in this case, like in some bacteria such as Clostridia, which has very elaborated systems to ferment amino acids, this pathway could actually, coming from threonine, produce a fermentation system that would reoxidize NAD and allows the metabolism to continue. And consistently, if you actually compare the ability of, let's say, if you do just mix wild type and a delta TDCB mutant, they have no difference in biofilm formation capacity, etc. And you mix monoculture experiment, you have no defect of this TDCB mutant. But if you mix these two bacteria, you have a very strong fitness defect, more than 90% fitness defect in this case. But if instead of inactivating TDCB, you inactivate TDCD, which has no impact on propanol production, then you have no difference in fitness between the wild type and the TDCD strain. And so suggesting that this pathway is actually provide a competitive advantage to bacteria that are able to do that. And if you mix actually an E. coli that has the pathway and an enterobacteria or another bacteria that does not ferment threonine, you also have a competitive advantage of the E. coli, whether or not it has the capacity to produce propanol. So this study had been mainly led by Sylvie Letoffe in my lab in collaboration with Franz Stressmann and Bianca Audrin and initiated by Sabina Chalabaev. And it shows, in collaboration with very good people doing mass spectrometry and NMR in two engineering schools in France, it shows that even looking at complex community formed by bacteria that have been studied for so many years now, you can still identify pathways or functions that have not been identified when studying them isolated and planktonic microorganisms. And so it can give us insights into what is going on metabolically when this culture and this lifestyle is actually developing. It can also give us hints onto new bacterial function, and besides this sort of molecule, you can also have other molecules, and we can hypothesize that it can have also communications molecules. It also gives us the possibility to think about metabolic engineering in a different way, so we can use some of these capacity to our benefit. It can also, it also means that there are some molecules that are almost exclusively associated or strongly associated to this lifestyle that could be used to detect it. And if, in some cases, when it's needed, pathogenic biofilm and so on, they could actually be the base of the development of biomarkers. And with this, I would just finish by, acknowledging the funding, and so the LABEX, IBEID, and also the Fondation pour la Recherche Médicale. And I would be very happy to try to answer your question.